All right, so I thought I would go through Chapter 12's problems, like all other problems. It's definitely good to, after watching a video or reading the book or both, to see if you can't answer these questions and do these problems and do them on your own. And then just look at the book when you need them, when you need to look at the answers. But don't look at the answers too soon. Try to do as much as you can first. All right, so here are some problems that I thought best represented the. I'm going to make this a little bit bigger, make it a little bit easier to. That might be a little bit too big. All right, so what is semi-conservative replication? All right, that is the accepted and well understood replication mechanism for DNA replication. And the whole thing is when you replicate, you get new DNA, and each new DNA has one old strand and one new strand. This is old, this is new. Semi-conservative uh, means one half of the, so semi is one half of the original molecule is present in new molecule. And just to throw the 5' to 3' on there, it's not really all that relevant right now in this case, but that's semi-conservative, that's all it means. Semi-discontinuous. I think I might have added this question because I kind of like it. Well, I made it up, so it's got to be good, right? What semi-discontinuous means is that when DNA replicates, one strand has to be discontinuous, but one strand can be continuous. And I think I drew this differently than I normally do, and we'll pretend I did it on purpose, but the importance of it is it doesn't matter which strand you use as the 3' and 5' strand, but once you decide to label one thing the 5' end and 3' end, then everything else has to be consistent with that. So all new synthesis has to be in the 5' to 3' direction. And so this 5' is relating to that end. And so this is the 5' and the 3' end of that new molecule. And here's the 5' to 3' end of that molecule. This is continuous synthesis. This is discontinuous. So I suppose you could call it semi-continuous or semi-discontinuous, but they tend to call it semi-discontinuous. It's kind of a nice big word to impress your friends. Semi-discontinuous synthesis. All right, and I kind of just drew that sort of. Dry molecule undergoing rolling. So we didn't do rolling circle. We didn't do a rolling circle, but what the hell? Give it a try. It's kind of interesting. So we got a molecule of DNA, and it undergoes a nick. And so it reveals that 5' end. And then the new DNA is going to come in and be extended off of this 3' end here. So it's going to come in this way and start to copy. And so there's our inside strand. Here's our 3' end. And it displaces the 5' end. So it displaces that 5' end, and then as the new DNA gets made, again, the same rules apply. It's got to go off the 3' end and go in the 5' to 3' direction. But as it does that, it displaces that 5' strand. And then eventually, this 5' strand is all displaced. And we're copying that, we're copying this guy. And this is our 5' end up here. Eventually we could put a primer on down here and copy that strand too, if we wanted to. And then eventually we'd get both strands copied. So that is the rolling circle model. I don't think we have Okazaki fragments and all that stuff in here. It's just kind of a continuously synthesized thing. And oh, I skipped one. Okay, so undergoing theta replication. So theta replication, that's the one that you really need to know. That's the one that's common. And make a replication bubble. So it looks kind of like a theta. Or it kind of looks like the big Ghostbusters Slimer ghost. And let's go with our primers being red. And let's say that the origin is here. And it doesn't really matter what direction we go, so as long as once we start we're consistent. So we're going to have a primer going that way towards the fork. Then we'll have to have a primer going that way away from the fork. On this side we could have a primer going toward the fork, and then here a primer going away from the fork. And we'll make our new DNA green. This is going in this direction. This is going in this direction. So it's going towards the fork that it's closest to, I guess. It's a leading strand. So this is leading, and that's leading. And then let's go a little bit later. We'll make a bigger bubble. Yeah, might as well try to keep the colors the same. So make a bigger bubble this time. And this one that was going away from the fork, well, that would have been, so our origin is here. And we would, so this is the primer that was already there. And it had already been extended towards there. But now we can add a new primer and extend it that way. This one that was going towards the fork can keep going. This one that was going towards this fork, it started there and it's going towards this fork. So it's still the leading strand, it can keep going. This one is going away from the fork in this case, so now it had to wait for this fork to open and then it could lay down a new primer. And we got some Okazaki fragments here. We got some templates. We got to label our 5' and 3' end. And label this 5' and 3' end. If that's the case, if this new strand on the top here is going 5' to 3', that means that blue strand on the bottom's got a 3' on, no, it's got a 5' on this end, 3' on that end, 3' there. Location of primers, we showed those. Template strands, new strands, leading strands, lagging strands. So this is leading, this one's leading, so that makes, I guess it's easier to see over here. This is lagging, leading, leading, lagging. So I think we labeled most things that we were supposed to label. Eukaryotic linear replication. So in this case, we're just going to have a bunch of bubbles. Primers, it's going to be a little hard to draw, but primers going towards the forks. And all these replication is going in both directions because it's bidirectional. So the fact that it's bidirectional just means that the bubble's opening up in both ways. So I think we need to make these bubbles a little bit bigger. And we'll just draw one of these. So we had this guy, primer towards the fork, a couple primers away from the fork, we got a primer away from the fork here, probably have a couple of them, and this primer is going towards this fork. So that's the general idea. What are the requirements for DNA replication? What I'm getting at here is that we need a template. Need a primer. Because we need a 3' OH. We need DNTPs. And we need a polymerase. Now we probably need ATP for energy too, but we haven't really talked too much about that. Template, primer, DNTPs, polymerase. That's basically it. Skip it over 7 somehow. Maybe I don't have 7 on here. Alright, we're skipping right to 8. The different proteins and enzymes take part in bacterial replication give the function of each in the replication process. Alright, so this is a good thing to just sort of memorize all these things and just go through this process a few times. So we need initiator, helicase, single-stranded DNA binding protein, DNA polymerase 1, DNA polymerase 3, DNA ligase. Oops, sorry, well, let's do 3 first. And then 1. DNA ligase. That's pretty much it. We need helicase. We need a gyrase. So this binds the origin. Kind of starts the process off. Unwinds DNA. Keeps it unwound. Major synthesis. And also proofreading. Which means 3' to 5' exonuclease. And 5' to 3' polymerase activity. This removes RNA primers and replaces with DNA. And it uses 5' to 3' exo and 5' to 3' polymerase activity and ligase. Connects Okazaki fragments. And we could probably expand on that a little bit, but that's the idea. Why is DNA gyrase necessary? So what this basically does, just in words, is relieves tension in front of the fork. So we got our fork like this. As this DNA gets unwound, it's being spun to kind of get unwound, and then you really just have to think of a rubber band. If you like have somebody hold one rubber band. So you take the rubber band and kind of twist it up, and then take the two two different strands of the rubber band and pull them apart. And as you do that, it's going to force the twisted part of the rubber band in front of that fork to get all tangled and then start to fold up on itself. And so we don't want that to happen. And so it kind of unwinds it. So even it gets wound up. If we didn't have helicase, this would get all kind of wound up like this. And helicase, or gyrase, I mean, unwinds it. So gyrase is an example of a topoisomerase. And what they do is they cut DNA strand and then kind of unwind it and then re-ligate it back together. All right. What similarities and differences exist in the enzymatic activities of DNA polymerase 1 and 3? What is the function of each? So, they both have 5' to 3' DNA polymerase activity. They actually both have 5' to 3' exonuclease activity. POL1 has, I'm sorry, this is 3' to 5', so this is the backwards one. So that's the one where we go 3', 5', 5', 3', and then we chew backwards to get rid of DNA so that we can replace it. POL1 has 5' to 3' exo activity. That's where we are coming up on the back end of a primer and we want to chew it up in the back and replace it with DNA. And so only POL1 has that. And so the function of POL3 is it's the major synthesizing enzyme. So DNA synthesizing enzyme. And it does proofreading. And POL1 removes the primer. And replaces with DNA. Why is primase required for replication? Because DNA polymerase cannot do, just to keep it simple, de novo synthesis. It requires a primer. And primase is a RNA polymerase and doesn't require a primer. And it can do de novo synthesis. And words and pictures how telomeres at the ends of eukaryotic chromosomes are replicated. All right. There's a 3' overhang that's left over after the last primer is removed. Telomerase extends that end by adding TT GGG repeats, or it could be TTAGGG repeats. And it does that using its own RNA template that has a template sequence, AACCC, and it just has that one copy of that, and then it keeps moving down and adding more and more repeats. And then after it does all that, and now we've got all these repeats that are added, now we can get just a let's make it a different color. Now we can just have some other primer, just by a primer from primase, come in, and then we can make the other strand of DNA. I guess we'll make it orange. And then we can have DNA ligase tie that up. So, again, we'll lose this little bit at the end. We don't care because we just added a bunch of this. So we keep adding more of that and losing a little bit, adding a little bit more, losing a little bit. And I would probably have to throw a few more words on there if I was answering this in terms of a test question, but I'm just I don't have this diagram handy. I could probably get it. Let's see. Following diagram represents a DNA molecule that is undergoing replication. Draw on the strands of the newly synthesized DNA and identify the polarity of newly synthesized strands, leading and lagging strands, Okazaki and RNA primers. Oh, let's see. I'm going to go to your ebook real quick. It's kind of nice having that whole ebook thing because I never have my actual book with me. Go into Launchpad. I could check up on you guys while I'm doing it. See how you've been moving along. But I think we've already drawn these things a few times, so I don't think there's going to be any surprises here. All right, so this would be in chapter 12, number 25. to the end of chapter 12 problems. Go down to chapter 25, or question 25. That's not question 25. I guess it's a little bit different in the some of those end of chapter problems are a little bit different, so I'll just go straight to the ebook. Chapter 12, go down to the end. And probably an application question and problem. Alright, so the diagram in the book looks something like this. Number 25 in the ebook or in the regular book. We've got our origin. Got our fork is unwinding in this direction. And this fork's unwinding in this direction. And the top strand is 3 prime. The bottom strand is it's like this. And so that's the key thing. So when we're going to draw our new strands, we have to make sure that they are consistent with those template strands that we just drew. So that means that there could be, if we're on this top strand, because this is 3 prime, that means that I can start my 5 prime to 3 prime primer right there, and that'll be the leading strand going towards that fork. Then the other DNA on this strand has to be going in the same direction. It still has to be going 5 prime to 3 prime in this direction. So they always have to go in the same direction on the same strand. And then the other one will be going in the opposite direction. And this one will be going towards the fork, so it will be the leading strand. And then this one will be have a couple of primers. Be going away from that fork, and so that'll be the lagging strand. 5 prime, 3 prime direction. This is 3 prime. So you got to kind of keep those things straight. So that whole new thing is going 5 prime to 3 prime on the bottom and 5 prime to 3 prime on the top. Okazaki fragments. RNA primer. Leading, lagging, and that pretty much does it. Which of the polymerases shown in table 12.3 have the ability to proofread? I'm not going to bother going to look at table 12.3. The main one I want you to know for this is DNA polymerase 3. And I want you to know what proofreading is. Oops. Then we have this enzyme is going to chew back in this direction until it gets past where it was. then it's going to replace that with new DNA. So that's proofreading, 3' to 5' exo activity, 5' to 3' polymerase activity. DNA PAL1 can do it as well, but PAL1 has its own special features that DNA PAL3 can't do. So just focus on the fact that DNA PAL3 can do that. Why might telomerase be a good target for cancer drug? Oh, did I get a little ahead of myself there? I must have skipped over this somehow. I'm sorry. I wouldn't think it would be this hard to navigate in Word. All right, so I'll go back to this one. What 3 mechanisms ensure the accuracy of replication in bacteria? The first one is there's proofreading. There's more than 3, but proofreading is done by DNA PAL3, and it's what we just talked about a second ago. It's when you do this backspacing business and then replace the DNA when there's been a mistake. Then there's mismatch repair. So you've got a mismatch, and after DNA replication is over, these enzymes come in and check out that DNA damage, and then they fix it, and they cut out the bad one, and then they replace it with a new one. And the other one's probably nucleotide excision repair. That's a little bit different. So this is a normal base. It's a mismatch, and so there's a special system for where the bacteria can tell which is the wrong base that's been put in. Nucleotide excision is when there's actually an abnormal or a damaged base put in there. And then nucleotide excision comes in and cuts out that bad base and then replaces it with new DNA. There's a variety of repair mechanisms, but those are the 3 main ones. How are these things different and similar? Well, they're similar. They need the same things. So they all need template, primer, DNTPs, polymerases, have to synthesize DNA in the 5' to 3' direction. So all the fundamental rules of DNA synthesis occur to both. What's different in eukaryotic replication is that there are multiple origins. Eukaryotic chromosomes are linear, so you have the end replication problem. And there are many more enzymes. It's just more complicated. They do the same basic things, but it's just there's more copies of each enzyme and it gets a little bit more complicated. How else is it different? Multiple origins, linear. Yeah, that's probably about the main things. Alright, so we've done this a few times, so I think you probably know the answer to this. What is the end replication problem? after you get, you end up with having a difficulty copying that end, and if you don't copy it, the chromosome gets shorter with every cell division. Let's try to make that a little bit better. So we'll just do it this way. We got a chromosome, gets shorter, gets shorter, gets shorter. That's in the absence of telomerase. If you've got telomerase will continue to add repeats onto the end and maintain the length of the chromosome. So that's how it does it. It's basic replication in E. coli. So if cells are dividing rapidly, that's cancer. That's a simplified version of cancer, but that's definitely part of what cancer is. And so cancer cells express telomerase. So normal cells don't necessarily express telomerase, but in order for cancer cells to continue to divide, they express telomerase, and so they maintain chromosome length, and so they don't die. They can keep dividing without dying. So these cells have telomerase in them, maintaining the chromosomes. And so if you have a drug, some kind of drug that targets telomerase and it inhibits it, it'll prevent it from lengthening the chromosomes, and it'll cause the chromosomes to get shorter. And then let's say you've successfully done that, and the chromosome now, when these cells divide, the chromosome gets shorter with each division. Eventually, this signals the cell to die. Die, die, die. So, if you can block telomerase, you at least, that's a potential target to cause dividing cells to die. Well, if you delete the RNA component of telomerase, it won't work, because then you won't have. You need to have a template to copy that. So it just won't work, and it's an essential part of the enzyme, and it won't work. And you won't have a functional telomerase. You won't lengthen chromosomes, and the cell will start to die after a few divisions. Challenge questions. How many stupid challenge questions? A conditional mutation expressed immune phenotype only under certain conditions, and expressed the normal phenotype under normal conditions. So yeah, temperature sensitive mutations are pretty common. So you might have like 25 degrees, have a normal phenotype, and at higher temperatures, let's say 30 degrees, and this is Celsius, you have a mutant phenotype. And these are useful because sometimes when you make a mutation, if it kills the organism, it's not very useful, because you can't get any organisms that have that mutation because they all die. So you can make them temperature sensitive, and then they only die at higher temperatures, and then you can sort of shift the organism to that higher temperature when you want to study the effect of that mutation. But I didn't really, let's see. So you have temperature sensitive mutations in all of these things. All right, so if we have mutants in these kinds of things, if we had DNA ligase and we were able to just sort of stop it, what we'd end up seeing is we'd see a bunch of DNA with a bunch of nicks in it. So we wouldn't be able to seal those nicks. DNA polymerase 1, we'd have DNA that still has all these primers in it, because DNA polymerase 1 wouldn't be able to come along and get rid of these primers. So we look like that. DNA polymerase 3, we would never be able to, we would have no replication. So none of these would be able to do anything. So this is kind of a dumb question. No replication. I mean, if you can't start it, you can't continue it. And I think that was the end. So we'll just leave it at that. If you don't have DNA polymerase 3, you can't copy anything. If you don't have primase, you can't initiate it. If you don't have initiator protein, you can't initiate it. So you would get no replication. All right, so that was a pretty quick go through on these problems, but you have the full written answers if you want them. I just wanted to give you my take on them very quickly. So we'll just leave it at that.